There are 15% of humans alive on this planet right now who carry a genetic anomaly that, by every rule of evolutionary biology, should not exist. Not rare, not unusual, should not exist. These people lack a protein that virtually every other primate on Earth naturally produces. Their immune systems reject what 95% of humanity accepts without question. During pregnancy, their own bodies can turn against their unborn children in a biological conflict so severe that, before modern medicine intervened, it killed infants across countless generations. And in isolated mountain communities, their numbers reach concentrations so extreme that the mathematics of natural selection simply cannot account for them. For decades, researchers kept discovering more questions than answers. The latest DNA research has finally revealed the mechanism. But the origins, the real, deep, uncomfortable origins of where Rh-negative blood came from and why it exists at all, remain one of the most genuinely mysterious problems in all of human genetics. What you are about to discover will make you reconsider everything you thought you understood about human genetic diversity, because something is wrong with the story we have been told. Not dramatically wrong, not in a way that shows up in headlines or press releases. Wrong in a way that sits silently in the background of medical textbooks and scientific journals, accumulating evidence year by year until the weight of it becomes impossible to ignore. 15% of humanity carries a biological trait that conventional evolutionary theory struggles to explain. They lack a protein that every other mammal in the rhesus family naturally produces. And for nearly a century, no one has definitively answered why they exist. These are the Rh-negative blood carriers. And what researchers have discovered about their origins in the last several years has shattered assumptions that stood for nearly a century. When Dr. Carl Landsteiner first identified the rhesus factor in 1937, working alongside Alexander Wiener, the discovery seemed almost straightforward. They had immunized rabbits with blood from rhesus macaque monkeys, and the antibodies the rabbits produced reacted with the blood of most humans. They called it the rhesus factor. The logic seemed clean. The protein was either present or it was not. 85% of people had it, 15% did not. Simple categorization, medical problem solved. The mystery was supposed to stay solved. It never stayed solved. The deeper researchers looked, the more the apparent simplicity collapsed into something far more troubling and far more interesting. Think carefully about what that absence actually means in biological terms. The Rh factor is a protein displayed on the surface of red blood cells. If you are Rh negative, your blood functions normally without it. The protein is not essential to your survival as an individual. The problem is not internal. The problem emerges the moment your blood encounters Rh positive blood, which is the blood of 85% of all humans. Your immune system sees it and responds the way it responds to any foreign invader. It recognizes the D antigen on Rh positive cells, identifies it as something that does not belong, and mobilizes against it. In a standard transfusion scenario, this creates manageable medical complications. But during pregnancy, the situation becomes something that evolutionary biologists have struggled to name without contradiction. They call it evolutionary roulette. Because an Rh-negative mother carrying an Rh-positive baby, a situation that arises naturally and regularly whenever the father carries the Rh-positive gene, faces a biological conflict that should, by every rule of natural selection, have eliminated this trait from the human population thousands of years ago. Here is how the conflict unfolds. When fetal blood crosses the placental barrier during birth or during miscarriage, or sometimes during trauma, the Rh-negative mother's immune system detects the D antigen in the fetal blood and produces antibodies against it. In the first pregnancy, the damage may be minimal. The immune response has not fully ramped up yet. But in subsequent pregnancies with Rh-positive fetuses, those antibodies cross back through the placenta and attack the developing baby's red blood cells directly. What follows is hemolytic disease of the newborn, anemia, jaundice, brain damage, death. This was not a rare historical tragedy. It was documented across centuries. Pregnant women died. Babies died. The same mothers who survived their first pregnancies lost subsequent children to a biological process occurring invisibly inside their own bodies. And yet, through all of that, through all of those generations of reproductive loss, the Rh-negative trait persisted. In certain populations, it did not merely persist, it increased. This is where the evolutionary puzzle stops being an interesting footnote and becomes a genuine scientific crisis. The answer that geneticists expected to find, when they finally had the molecular tools to look, was a simple one: a single mutation event arising in one ancestral population, spreading through migration and founder effects, concentrating in isolated groups through genetic drift. A clean, standard evolutionary narrative, the kind that fits neatly into textbooks. What they found instead was something that required the textbooks to be rewritten. To understand the full weight of the discovery, you first have to understand what the Rh factor actually is at the molecular level. And this is where the story takes its first genuinely unexpected turn. The Rh factor is produced by a gene called RhD. In Rh-positive individuals, this gene is present and functional, producing the D antigen protein that appears on red blood cell surfaces. In Rh-negative individuals, the RhD gene is not simply mutated or switched off, it is gone. The entire gene has been deleted from the chromosome. This is not a small change. This is not a single base pair substitution. This is the complete removal of a gene that the vast majority of humans carry. And when researchers examined the molecular mechanism responsible for that deletion, they found something architecturally remarkable. Flanking the location where the RhD gene should be are structures called rhesus boxes, identical 9,000 base pair DNA segments that share 6% genetic homology with each other. These near-perfect mirror sequences create a specific structural vulnerability. During cell division and reproduction, these sequences can misalign. When they do, the RhD gene sitting between them gets excised from the chromosome entirely. What remains is a hybrid rhesus box, a molecular scar marking where the gene once was. The deletion is clean, it is precise, it is structurally repeatable, and it is mediated by a specific molecular architecture that researchers have now mapped in extraordinary detail. But here is where the story breaks open in a way that no one anticipated. Recent studies examining Rh-negative populations across multiple continents, Iran, China, Thailand, Saudi Arabia, Sweden, and across Western Europe, discovered something that does not fit any simple evolutionary narrative. The deletion mechanism is not the same everywhere. In European populations, particularly among Caucasians, the dominant mechanism is the complete RhD deletion mediated by the hybrid rhesus box exactly as described. But in East Asian populations, the primary mechanism involves something geneticists call Deal variants, weak D genotypes, in which small segments of the RhD gene remain present and partially functional, producing minimal D antigen expression rather than none at all. And in African populations, additional mechanisms including pseudogene formation and specific point mutations create yet another distinct genetic background for the same observable result. The same blood type, Rh-negative, produced by fundamentally different genetic mechanisms in different populations around the world. Read that again slowly, because the implications are staggering. If the Rh-negative trait had arisen from a single ancestral mutation spreading through the human population via migration, as simple evolutionary theory predicts, then all Rh-negative individuals should share the same underlying genetic architecture. The mechanism should be the same everywhere, because it should trace back to the same original event, but it does not. The mechanism varies. The genetic pathway differs. The molecular history is population-specific. This means the Rh-negative trait did not emerge from one place and spread globally. It emerged multiple times, independently through different genetic pathways in isolated populations, separated by geography and tens of thousands of years of time. The same phenotype, the observable absence of the Rh factor, evolved convergently from completely different genetic starting points in different corners of the human world. In 2026, research from the Chongqing Blood Center documented 15 years of Rh-negative donor data across Chinese populations. The results showed dominant genetic configurations completely absent from European populations. The CCDE haplotype, one specific genetic arrangement, appeared in over 50% of Chinese Rh-negative donors. The same configuration is virtually non-existent in Caucasian Rh-negative populations. Simultaneously, the European-type genetic background is rare among Asian Rh-negative individuals. Two populations, both carrying Rh-negative blood, separated by the entire span of human migration history, carrying entirely different genetic architectures underneath the same observable trait. The implications reach far beyond blood typing. If independent genetic events produce the same blood type in different isolated populations, then human genetic diversity is operating on a level of complexity that conventional models were not designed to capture. The standard picture of human genetic history, mutations arising in a common ancestor, spreading through populations via migration and natural selection, is not wrong exactly, but it is radically incomplete. It misses an entire dimension of how human populations develop genetic distinctiveness, and then the ancient DNA research added another layer, one that the scientific community is still processing. Studies examining ancient DNA from Neanderthal specimens produced an unexpected finding. Neanderthals tested positive for partial D antigen expression. Not complete like most modern humans, not absent like Rh negative individuals, partial, intermediate. A configuration that sits between the two states that modern humans occupy. This suggests that our closest evolutionary relatives possessed an Rh configuration, distinct from both modern Rh positive and Rh negative humans. The Rhd gene was present in Neanderthals, but expressed differently. The molecular landscape was different. What this means for the origin of the Rh negative deletion is still being debated in the scientific community, but the timing evidence points in a specific direction. Analysis of the genetic structure surrounding the Rhd deletion suggests the event is relatively recent in evolutionary terms, emerging within the last 50 to 100,000 years at most, and possibly more recently than that. Some researchers propose the deletion occurred primarily within the last 50,000 years. This timeline places the emergence of Rh negative blood squarely within the period of the Upper Paleolithic expansion of modern humans into Europe, coinciding with the period when human cultural complexity accelerated, when art appeared, when population alongside the geographic distribution, and the picture becomes genuinely revealing. Rh negative blood appears at high frequencies only in specific regions of the world. The Basque country of northern Spain and southwestern France, where frequencies reach 35% in some studies and approach 40% in isolated valleys. Parts of Scandinavia, approaching 30% in some populations. Isolated communities in Ireland and Scotland, running between 25 and 30%. And then virtually everywhere else, most of Asia, most of Africa, most of the world, below 5%, sometimes close to 1%. This is not a random distribution, it is a geographic signature. And when you overlay it against what we know about ancient population genetics, the pattern locks into place with uncomfortable precision. The populations carrying the highest Rh negative frequencies are exactly the populations that retained hunter-gatherer ancestry longest, resisting the demographic waves that reshape the rest of humanity. The Basques never adopted Indo-European languages. They never absorbed the genetic signature of the Neolithic farmers who swept across Europe from Anatolia, or the Bronze Age, steppe herders who arrived from the east. They remained genetically and linguistically a world unto themselves. The Scandinavian populations, showing elevated Rh negative frequencies, contain some of the highest proportions of ancient European hunter-gatherer DNA anywhere on the continent. The Irish and Scottish populations with above-average Rh negative concentrations show greater genetic continuity with pre-Indo-European inhabitants than most of mainland Europe. What you're looking at in the geographic distribution of Rh negative blood is a map of ancient survival. A map of the populations that geographic barriers protected from the successive waves of migration that genetically overrode most of Europe and Asia, the mountain ranges that made the Basque country a natural fortress, the ocean distances that insulated Ireland and Scotland, the northern latitudes that sloped. And crucially, the Basque case demonstrates something fundamental about how genetic traits accumulate in isolated populations. In the Pyrenean communities where Rh negative frequencies approach 40%, the reproductive costs of Rh incompatibility should have been devastating. Mothers attacking their own fetuses across generations, losing children to hemolytic disease at rates that should have created severe reproductive bottlenecks. And yet the population did not collapse. The genetic frequency did not decline toward the global average. The trait stabilized and in some communities increased. How did this happen? This is the question that conventional evolutionary theory cannot cleanly answer. The possibilities that researchers have proposed include several directions, none of them fully settled. Some propose that Rh negative status carried selective advantages under conditions specific to the ancient environments these populations occupied. Perhaps resistance to certain parasites or pathogens that were prevalent in those regions during specific periods. Some propose that the social structure of these isolated communities, marriage patterns, kinship networks, cultural practices, created demographic conditions that mitigated the reproductive cost of Rh incompatibility more effectively than we currently understand. Some have speculated about whether ancient populations might have developed empirical knowledge, specific birthing practices, postpartum protocols that reduced infant mortality from hemolytic disease without understanding the molecular mechanism behind it. None of these explanations is fully established. All of them point toward something important. The persistence of Rh negative blood in certain populations is not explained by the genetics alone. It required something social, something behavioral, something about how these communities organized themselves across time to survive the biological pressure that should have eliminated the trait. The most recent molecular research has also revealed health implications that go beyond pregnancy complications in ways that mainstream discussion rarely addresses. Studies correlating Rh negative genotype with autoimmune disease prevalence show patterns that vary by genetic background. European type RHD deletions show different autoimmune associations than East Asian DEAL variants. The same absence of the Rh factor produces different physiological consequences depending on which other genes surround it, which population carries it, which genetic environment shaped its evolution over generations. This is where the research is moving right now in 2025 and 2026. Away from simple phenotypic classification, you are either Rh negative or Rh positive, toward molecular complexity that recognizes Rh negative populations as genetically heterogeneous despite sharing an identical blood type designation. The same label applied to fundamentally different genetic architectures in different people from different populations. And this distinction has immediate practical consequences that most people carrying Rh negative blood are entirely unaware of. Research published from Eastern Iran in 2026 established that hybrid rhesus box analysis now serves as a reliable molecular marker for accurate RHD typing. An individual carrying the weak D phenotype, partially positive for D antigen, may test negative through standard serological screening, but carry partial RHD function. During transfusion, this creates risk scenarios that conventional testing cannot detect. The person appears Rh negative on the standard test. They receive blood products matched for Rh negative individuals. But the partial D function in their cells creates immune interactions that a fully negative individual would not experience. This is not a theoretical risk. Blood banking institutions worldwide are currently transitioning toward molecular genotyping protocols because serological testing alone is no longer sufficient. The discovery that different genetic mechanisms underlie the same blood type designation in different populations forced a fundamental revision of how blood banks identify and classify their donors and recipients. If you are Rh negative and you think your blood type is a simple binary, present or absent, the science that emerged in the last several years suggests your situation is more complex than that classification captures. The geopolitical dimensions of this research are being actively managed in ways that deserve attention. The studies that produce these findings required international collaboration between blood centers in Iran, China, Thailand, Saudi Arabia, Sweden, and Western Europe. No single nation's research could answer the questions alone. The Chinese population data revealed genetic mechanisms absent from European reference databases. The Iranian research identified patterns distinct from Middle Eastern neighbors. The Thai studies distinguished East Asian patterns from Southeast Asian ones. What has emerged from this international coordination is a global genetic map of human diversity that challenges every simplified model of human evolutionary history. It also means that molecular blood typing now generates something that did not exist before. A genetic fingerprint that identifies not just blood types, but population ancestry. An Rh negative individual can now be molecularly characterized as carrying the European type complete deletion versus the East Asian DEAL variant versus the African pseudogene mechanism. These are genetically distinct categories that correlate with population ancestry in ways that go well beyond medical utility. The infrastructure of blood banking worldwide is quietly becoming a system for documenting human genetic diversity at population resolution. The implications for privacy and for the ethical use of population-level genetic data are underexplored and underregulated. But set those concerns aside for a moment and return to the deeper question. The one that started all of this: why does Rh negative blood exist at all? The answer that the research is converging toward, incomplete, still developing, but pointing with increasing clarity in one direction, is that Rh negative blood represents multiple independent evolutionary events occurring within the last 50 to 100,000 years in geographically isolated human populations. It is not one story. It is several parallel stories that happen to arrive at the same phenotypic destination through completely different routes. In European populations, a specific molecular accident, the misalignment of rhesus box sequences during cell division in one or several ancestral individuals, produced the complete RHD deletion. Those individuals survived. Their descendants survived. In the geographically isolated communities of the Pyrenean Mountains, the ancient hunter-gatherer territories of Scandinavia, the ocean, bounded communities of the Atlantic fringe, the deletion accumulated to high frequencies because migration did not constantly introduce Rh positive alleles to dilute it. The trait concentrated not because it was advantageous in any clearly identified way, but because isolation allowed genetic drift to operate without dilution for long enough to push frequencies far above global averages. In East Asian populations, a different molecular pathway produced the DEAL variants, partial deletions with different structural characteristics and different phenotypic subtleties. In African populations, pseudogene formation and point mutations created yet another genetic architecture underneath the same observable result. 3 populations, 3 genetic mechanisms. 1. Blood type. This is the discovery that changes everything about how we understand the Rh negative story, and by extension, how we understand human genetic diversity more broadly. The lesson that Rh negative blood teaches, the lesson that is still being absorbed by population genetics as a discipline, is that human evolution does not always operate through single mutation, events spreading from common ancestors. It sometimes operates through convergence, through independent events in isolated populations, producing the same outcome through different paths, through geographic barriers, concentrating traits to frequencies that natural selection operating in mixed populations would never produce, through cultural and social structures that modify the apparent cost of biologically expensive traits in ways that extend their survival far beyond what molecular data alone would predict. The 15% of humanity carrying Rh negative blood are not the product of a single ancient genetic event preserved across the millennia. They are the product of multiple recent genetic events, concentrated in specific populations by specific geographic and demographic histories, producing a shared phenotype through fundamentally different molecular mechanisms. They are not a single story. They are several stories that rhyme. The questions that remain unanswered point toward research frontiers that molecular science has barely begun to explore. Why did the RHD deletion appear when it did within the last 50,000 years, during the most culturally and demographically dynamic period in human prehistory? What selective pressure, if any, accompanied its emergence? Were there disease resistance trade-offs in the ancient environments where isolated populations first accumulated the trait? Did the social structures of those populations, marriage patterns, kinship obligations, birth practices, create conditions that allowed the biological cost of Rh incompatibility to be absorbed without eliminating the carriers? Could there be immune regulatory advantages to Rh negative status that modern medicine has not yet identified, operating in ancient disease environments that no longer exist? These questions are not resolved, but they are being asked now with more precision, more data, and more sophisticated molecular tools than any previous generation of researchers possessed. And the answers, when they come, will almost certainly reveal that the story of Rh negative blood is stranger, more complex, and more illuminating about the full nature of human genetic diversity than either the simple medical classification or the century-old assumption of a single origin ever suggested. If you carry Rh negative blood, you are not carrying an anomaly. You are carrying evidence of a specific kind of human history, a history of isolation, of geographic barriers that preserved ancient genetic configurations while surrounding populations changed, of relatively recent molecular events that concentrated in communities that stayed put long enough for drift to do its work. You are carrying a genetic signature that identifies not just your blood type, but your ancestors' relationship to the waves of migration and mixing that built the modern human world. The populations with the highest Rh negative frequencies are the populations that most successfully resisted absorption into the demographic flows of the Neolithic and Bronze Age. They stayed, they maintained, they accumulated. And what they accumulated is now one of the most scientifically revealing markers of human population history available to researchers. The origin of Rh negative blood remains partially mysterious. The mechanism is now understood better than ever before. The timeline is clearer. The population-specific pathways are mapped. But the full story, why this specific deletion appeared when it did, what conditions favored its retention, what advantages may have counterbalanced its reproductive cost, is still being written. What is no longer mysterious is this. The 15% of humanity who carry this trait are not carrying a single shared heritage from a common ancient ancestor. They are carrying parallel genetic innovations from multiple isolated human communities, converging on the same observable outcome through different paths, preserved by the same force, geography, isolation, and time. The mountain protected what the migration would have erased, and what survived inside.